Pleasure to introduce today for this talk speaker, Michael Shadlen. Mike doesn't need any introduction, but just for the sake of formality, I say a few words about him. Mike went to Brown University for his bachelor degree and medical school there. And while he was at medical school, he did a PhD at Berkeley. Just remember that how people used to work there back then. After his PhD, he did a residency in neurology at Stanford, and after that, he joined Bill Newsom's lab to do a postdoc there. And after that, he moved to Seattle to start his own lab there. And after, I think, maybe 20 years or so, he moved from Seattle to Columbia to join the Zuckerman Mind Brain Institute. All of us know Mike's work quite well, and I know that people at IPM are following his work quite closely. I wanted to say something which I forgot. It's kind of very bad. Okay, I remember. Mike is also a good jazz guitar player, and I'm pretty sure in the panel he's as happy to answer your question about jazz and music as he's going to be happy to answer your neuroscience questions. And the third thing that I learned from Mike in his lab was that he tried very hard to teach us that we are as humans, and as humans we are responsible for the people that are less fortunate than us, people that are marginalized, and people that are discriminated against. And he lives by this example. When he was quite a young medical student, he went to Honduras to help people there. And since I know him, he's quite fighting for the people that are less powerful than most people. It's a great pleasure, Mike, to welcome you back to IPM after 16 years. And it's unfortunate that it's not in person, but I hope soon we can have you back here in full flesh. The floor is yours, Mark. Thank you.
Kheli Mamnoon, Mehdi. Shab Becher, everybody, and So Becher, I guess, here, if anyone's in the States. Good morning. Can you see my slide?
Yes, we can.
Okay, good. All right. So I'm going to feature today the work mainly of a very talented graduate student, Gabe Stein, and I'm going to talk to you about the two areas in the brain that are connected by a synapse. And it turns out that one of them represents the accumulation of evidence for a decision, and the other forms the termination of that decision. Okay, so we study, just some background, we study perceptual decision making because we think of it as a window on higher brain function. You know, the philosopher Plato used the term carving nature at its joints. I think decision making carves cognition at its joints and will ultimately teach us the building blocks of normal cognitive function, I think, hopefully, so one day we can repair them in disease. That's sort of my view as a neurologist and neuroscientist that guides the work. Now, over the years, you know, we've learned lots of things about perceptual decision making. We've learned about inference, and which we'll talk about today, and the integration of many samples of evidence over time, how we control the speed versus accuracy trade-off, how we assign confidence in a decision. That was work from Ruzbe Kiani initially, and how we direct our attention, and also how we even change our mind. And so I'm going to spend most of the day today worrying about these two problems, integrating evidence and effectively controlling the termination of decisions. Now, many years ago, Bill Newsom and Tony Movshin and Ken Britton began studying perceptual decisions by training monkeys to decide between the net direction of moving dots. And I'm sure most of you are familiar with these and maybe tired of seeing them. I know the people who review my grants are very tired of seeing them. But the monkey has to decide the net direction of these dots, and I don't know if you can tell, but these dots have a weak tendency to move to the right. We control the difficulty by controlling the probability that a dot plotted at one moment in time is displaced in motion the next moment, 40 milliseconds later. And sometimes we also control that probability. I'll call a motion strength or a coherence. And sometimes we also control difficulty by controlling how long the monkey gets to view these dots. But in these experiments I'm going to talk about today, the monkey controls the termination of the decision. He controls how long he views the dots, and he makes an eye movement, a saccade, a fast eye movement, to indicate what he thought the answer was. Like here, they thought the answer was the right choice target. And we like this what's called a choice response time or choice reaction time experiment. We like this because it provides insight into the speed versus accuracy trade-off, and it identifies, for the physiologist, it's very useful because it identifies the epoch of decision formation on each individual trial. Now, over the years, like 20 at this point roughly, we've learned a lot about this process, and I'm going to summarize it at a computational level here, which is that noisy evidence from the world, in this case the video monitor, gets transduced by neurons in the visual cortex and extrastriate visual cortex into direction signals. The main part of the visual cortex is area MT or V5, contains 90% direction selective neurons, and it's basically crucial for the monkey to perform this task. I'm showing you this evidence and calling it momentary evidence because it comes and goes with the video frames. And I've represented it as a Gaussian on its side, with a positive mean to indicate that this motion is rightward. So there's a net sense that the evidence has a likelihood of being positive, which we'll call rightward. The idea, though, is that neurons with longer time constants for computation, those that actually exhibit persistent activity, have the capacity to accumulate in their firing rates the momentary evidence. So this is the integral of signal plus noise, okay, what would be called drift plus diffusion, or a biased random walk. And this would be one particular example that ends in this upper absorbing bound, okay? And that bound says, I've had enough information, I'm going to answer right. And of course, there's a lower bound down here, and I'll qualify this lower bound in a moment. But the idea then is that we can unite the notion of what decision the monkey makes, or we, because we behave just like monkeys on this task, that is what decision, right or left, with the amount of time it takes to reach it. And just to get an intuition for the speed versus accuracy trade-off is that if these bounds were set closer to the initial starting point of the accumulation at 0 here, you'd see this trial would have ended with a leftward choice, which would have been an error. But of course, there would have been a savings in time. Okay, now if you look at data from humans or monkeys, this is monkey data, and you plot the reaction time against the signal strength, the motion strength, you get these points. So reaction time is the time that elapses from onset of the random dot motion to the beginning of the monkey's eye movement response. Okay, and this motion axis is signed so that strong rightward motion are the big positive numbers, strong leftward motion are the big negative numbers, and 0 in the middle of the graph is when the motion is completely ambiguous. Okay, there's information in the stimulus, but it's on average it doesn't favor right any more than it favors left. And at that point, you see that's when the reaction times are the longest, nearly a second. And they fall off to numbers that are on the order of 400 milliseconds or less at the easiest conditions. Now, this very simple depiction of sort of optional stopping of an accumulation of sequential samples of momentary evidence is solvable, and the curve I've just drawn is a fit to of a very simple fit of a very simple model that would be the time t that a decision would terminate, okay? Now the reaction time, which is really what I'm representing here by t, gets another addition, which is there are some part of the reaction time is not accounted for by the decision, so I'll call that the non-decision time. And then you see I already introduced this quantity b, this variable b, that's the bound height, and it controls the speed accuracy trade-off, and there's only one other degree of freedom, which is this constant k that multiplies the motion strength, uppercase C. Okay, and that just converts our motion strength terms, which I call a probability or a coherence, it converts it into a signal to noise. Okay, and that's all you need to solve for the stopping times and then add a constant for the observed reaction time. Now, the very interesting fact is that this same, as I said, this same process also explains the likelihood you would end in this bound before this bound. So that gives rise to a choice function. But the interesting thing is that the choice function contains the same two free parameters, the k term and the b term. It's a logistic, actually, which is very deeply interesting. I think it explains why under the hood we're basically rational. We accumulate in units of log likelihood ratios to make decisions. Okay, that's a quick aside, and you can make me elaborate on that later if you like. But the point I want to make with you here is, having established these constants k and b to explain the reaction time, we're in a position where we predict that the motion strengths on the far end here are going to lead to 100% rightward choices, and the ones over here are going to lead 100% leftward choices, that is, no probability of choosing right, and then the rate of change of that choice function will follow this dashed curve, and that's pretty much where the data lie. Okay. Now, I should just say that this way of making decisions, where you want to take sequential samples and probably use as few of them as possible to achieve some desired level of accuracy, is the way that one of the tricks that Alan Turing used to break the German Enigma code in World War II, and it's also the foundation of the mathematics was also developed during World War II, also in secret work here at Columbia University by a mathematician named Abraham Wald. That's for your amusement, and we can talk more about the code breaking if you like. I love telling that story, but it's a little bit off-piste, and I won't get through the science if I tell you now. So okay, so let's talk about the neural mechanisms briefly, just again in the form of review. When I started this work with Bill Newsom, I was a postdoc in his lab, and he turned me on to this project. We thought, okay, these neurons have to receive information from direction-selective neurons, directly or indirectly. They have to project to the oculomotor system, directly or indirectly. And they should exhibit these long time constants, so that they could represent not the momentary evidence like a sensory neuron keeping up with a changing world, but like a neuron that would have some persistence and therefore the possibility of representing an integral. Okay, and so over the years we've looked at lots of places in the brain. I'm going to talk to you about two of them today, the lateral intraparietal area, which has been the most charitable to us in terms of revealing secrets of how things work, but it's not unique. And I'm also going to talk about the superior colliculus, which we thought played a role like LIP, but it turns out we were wrong. Okay, so here's here's a picture of the monkey brain, and we'll look at a coronal section, through the brain at this level. This sulcus you see here on the two sides of the brain is the intraparietal sulcus, and the area, the lateral intraparietal area, LIP shown here in orange, is the area that we're going to focus on. This was an area identified by Richard Anderson as the part of Brodmann's area 7 that projects to the frontal eye field and the superior colliculus. So, we think of the parietal cortex as involved in knowing things, like when you lose it, you get agnosia. But really, we should think of this area as taking information and assigning meaning to it for purposes of some kind of action in the world, what J.J. Gibson would call an affordance. In this case, the affordance is grasping with the gaze. Okay, all transient things in the visual world invite us the possibility of looking at them. That would be the use of the term affordance in Gibson language. Okay, it's a very by the way, that point of view, the way of thinking about the brain in this sort of intentional as opposed to a representational sense is very rewarding. I'd say it's been probably the secret to any success I've had in my career. It's because I don't think it's wise to be decoding information in the brain and putting labels on it. In other words, working on neural networks and machine learning classifiers and stuff. But rather, the point of the brain is to figure out what it might do with that information. Okay, so now back to the theme here is that I want to illustrate what spatially selective persistent activity is, because this is the hallmark of the LIP neurons that interest us. Okay, and here you'll see a monkey make an eye movement to the remembered location of that briefly flashed red spot. I hope it showed up. Sometimes it does and sometimes it doesn't on these video presentations. Okay, the monkey doesn't see this gray. That's for you to know where the red light flashed, okay? And that's going to turn out to be the response field of this LIP neuron. And hopefully you'll be able to hear, I think through my microphone, the spikes from a neuron while the monkey performs this very simple working memory task. Did you hear that? I don't know if anyone can even answer me, so I'll just assume you heard it.
Yeah.
You could hear it. Thank you. Let's see. I'll make it a little louder just in case that helps. Okay, so this is the persistent activity. Okay, this is the light came on and off. This is about a 2-second period where he has to remember its location, and then he's instructed to make an eye movement. And there's a latency, and he makes the eye movement. Okay, so now let's flash the target over here. Oh, that didn't work. Okay. Okay. I just wanted to say that persistent activity holds the key to higher brain function. I think it holds the key to higher brain function because it allows us to have thoughts in a time frame that's independent of immediate change in the world or the real-time demands of controlling the body. And I like to refer to that as freedom from immediacy. And I'll bet you that many of the breakdowns in higher brain function interfere with this capacity to hold information for some little period of time that connects the recent past to the immediate future. So here's what I wanted to show you now, another movie of the neuron, but now this demonstrates its spatial selectivity because we'll flash the spot over to the left and... Okay, so hopefully what you heard and now you see are the spikes, that basically went down, okay, back to background or maybe even a little below it while the monkey planned an eye movement to this other location. Okay, so what Bill and I decided to do, Bill Newsom and I, was give the monkey a reason to recruit these neurons. He's going to make an eye movement here if he thinks motions to the right and here if he thinks motions to the left. So all we've done really is to study decision making, is exploit a property of these neurons. And actually, we were very worried that what these neurons would tell us is, yes, the monkey has decided, has completed his decision and plans a rightward movement, or he plans a leftward movement, in which case we should be able to tell that by the firing rate. But we learned a lot more than that, and this was the very first reaction time experiment in the lab done by Jamie Reutman, and what you're seeing here are average firing rates just during the first portion of the decision formation. It's while the monkey's forming his decision. We know that because he hasn't made his eye movement to tell us that he's done. And what you see here on the solid curves are trials that are going to end with the monkey choosing the target in the response field. I'll just say choosing rightward, okay? And what you see in the dash curves are the leftward choices in the end, right? But he hasn't made up his mind yet. He's starting to do that during this period of time. And what you can see is these responses don't just reflect an all or none eye movement to the right or to the left. They represent something about the quality of the evidence because the gold curve is the strong motion. The blue curve, the monkey might as well have been guessing, but there are fluctuations even when there's just noise that push the monkey one way or the other. And then red is just one of the intermediate motion strengths. Okay, so this is telling us more than just what eye movement the monkey's making. It looks like it's at least coinciding with the evolution of that decision. And sure enough, at the end of the decision, when we align the same neurons, same spikes, but we align them to the time of the eye movement, we see that the responses have come together. And we can see that much better if we take all the trials and instead of grouping them by motion coherence, we group them by, whoops, I missed something. Darn. Oh well. I'll show you later that there's a better way to look at this graph, okay, which would show us that they really come together about 80 milliseconds before the saccade, and that's going to be very important later when we get to the termination mechanism. I do want to draw your attention to the fact that the responses don't come together when the monkey makes the opposite choice, and the reason why is because there's another set of neurons that accumulate not in favor of right and against left, but in favor of left and against right, and they hit an upper bound, and and whatever hits the upper bound stops both processes. Now, I want to now take a little look at what we think is going on during decision formation. Now, I showed you the responses. I want to reconcile the computational view, which is the accumulation of noisy evidence that can lead to an upper or a lower bound, okay? Or let's put it this way, that can lead towards a right choice or against it. That is, it would go negative. relative to the starting value. And I want to reconcile that with the curves I just showed you, which were conditionalized on the choice the monkey make. I only showed you in solid the ones that ended in this choice, and in dashed the ones that ended in this choice. But this is what the responses look like before you conditionalize on the choice. Okay, and what they look like is just the accumulation of something that might be noisy, but we've taken averages, so we've suppressed our representation of the noise. There barely wiggles in these curves, and what we see is the deterministic component driven by the motion strength. So here's the strong motion to the right, weak motion to the right, weak motion to the left, strong motion to the left, and this gray is the noise-only curve. Now, in our minds, what we think is going on individual trials is something more like this. So this would be a set of individual trials for the noise-only condition, because what we think is going on is that these averages that we show are averages of diffusion paths, okay? Now, that's been something that's kind of in our mind, and we've had indirect ways of describing that in the past. In fact, it's been part of a controversy, between me and the next speaker. But I'll show you very soon that we now have a handle on that. Here's the curve I wanted to show you that I thought was on the previous slide, which is when the responses end, okay, we can, by again looking at averages, but now grouping by quantiles of reaction time, now we get to really see that there's a critical time when it looks like something is probably stopping the process by reading off something like a threshold on the firing rates in the LIP, I don't know, somewhere like 55 spikes per second or something, maybe 60, okay? And I shouldn't say LIP because a lot of areas of the brain do this, so it's LIP or another area that's highly correlated with what's going on in LIP. Now, because we don't see evidence for that bound, as I said, when the monkey chooses left, I already explained this to you. What's really going on in the brain is a race among diffusion processes. And by saying they're diffusion processes, what I mean is that the right minus left can take negative and positive steps. It really looks like diffusion, okay? But what terminates that is an upper bound. And then there's another process, for the most part on the other side of the brain, that accumulates left minus right, okay? And of course, if these were perfectly correlated, and they're very highly correlated because they see the same motion information, then they would be mirror replicas of each other. We can draw them on the same graph and put an upper lower bound, and that works fine for the math, okay? But in reality, they're not perfectly anti-correlated. Okay. Now, LIP is part of a network of association areas, and here's just actually a fraction of that network, but probably the most important parts, or the more important parts. And I'm going to focus today on the two areas that define LIP. It's primary projection, so the frontal eye field and the superior colliculus. Okay. Actually, I'm going to focus on the superior colliculus now today. Now, so both of them exhibit spatially selective persistent activity, and they're thought to play a role in decision making, okay? And you'll see why in a second when I show you some slides. But our working hypothesis is that one or both of these play a role in decision termination. So we set out to test that, and the person who really led the charge in this work is Gabe Stein, whose picture you saw on my title slide. And what Gabe wanted to do was use this choice reaction time design. Of course, if you're really interested in understanding when the monkey terminates, that's a good thing to do. We had access to these neuropixel probes. Actually, we've been testers. The Howard Hughes Medical Institute put some money into this to move these neuropixel electrodes into the primate, which requires a lot of engineering, and it was pretty difficult to pull off. But we've been testing these over the years, and I'm going to show you why they're useful to us. Because essentially we really needed to do multi-neuron recording simultaneously from both of these areas. And Eric Troutman, who I want to introduce, was the person who really got this technology working in our lab. He was a former student with Krishna Shenoy, and he got it working there, and now we're working on lots of projects together. So this helps us in two ways. They allow us to record from LIP neurons and superior colliculus neurons that share the same response fields. Okay, so for example, that's a tricky thing to do, because if you have neurons that happen to have superior colliculus neurons, say, that has this response field, maybe a group of them that do. Okay, there's a map. They're tightly clustered, so that's easy. But there's no guarantee that you'll get LIP neurons that have this response field. So the way that we go about doing it is by using these probes. Now, these probes have 4,000 sites. 384 are active at the same time. We typically get 80 to 200 neurons, isolated single neurons, and on these probes. And of that, we get somewhere between 5 and 20 neurons that we would have that have relevant response fields. And relevant in this experiment means they overlap the same target as the one that's in the superior colliculus response field. Okay. Now, the other thing we get out of this, and I'm going to spend a little time on this today, is that we get so-called population spike rates. Let's just call, I'll call them mini population when I remember to. It's a subpopulation, because we get a bunch of neurons that share the same response field, and so we can then measure spike rates on single trials from the averages of those neurons. Okay. So here's one experiment. This happens to be just 7 LIP neurons that share the same response field as the colliculus. We had 13 superior colliculus neurons in this recording. And this is what the average firing rates look like. They don't look quite as good as the picture I showed you a moment ago from Jamie Reutman's data, but we were averaging many neurons there and many, many, many trials. So here's just one experiment, but you get the idea that it looks similar. Again, we see strong leftward motion, strong rightward motion. You'll see why I have left and right flipped on this one. And this is the noise only condition in the middle. And sure enough, I think I have a nice graph of that. If we now, instead of plotting response averages over neurons and trials, if we take single trial average over neurons, lo and behold, what we're looking at are things that look to the eye like diffusion paths, okay? So each one of these traces is the firing rate average of 7 neurons during a single trial, okay? And by the way, if you look at the variance of these responses, these single trial means over time. So the variance starts out small and it increases linearly for a while, and that starts to cap off. That's because of the presence of a bound. But that linear increase in variance as a function of time is a signature of diffusion, right? And you all know this because the sum of independent random numbers, random samples now of evidence, right? The variance of that is the sum of the variances. Now I want to make a little digression because this is a big thing for my field. So for a long, long time, neuroscientists have practiced what I call the ergodic assumption of the neurophysiologist. And that is they believe that they gained insight into the signaling of a single neuron by taking the average of that neuron across repetitions of some kind. And by taking those repetitions, they think, well, that's kind of like having a lot of neurons, like the one I recorded, and its activity, the average of those lot of neurons is like my firing rate as a function of time. So for neural computation, in order to in fact have any notion of a firing rate as a function of time and not some latent process that lives inside of a neuron and only gets out through the spikes that it emits, that in order to actually compute with rate as a function of time, one needs to have a lot of neurons, because otherwise you just have spikes and gaps between them. And those of you who've read my papers know that that argument leads to a whole set of conclusions about why neurons are noisy and why there's inevitability of correlation and on and on and on. But so I wanted to say this is actually a different session where we had more LIP neurons. It's not the one I just showed you. These average firing rates, like what we would do from single neurons, just averaging across the trials and so forth, they look a lot better. They look kind of like what we might see if we were averaging from 100 neurons. This happens to be 18 neurons, okay? And here they are coming together before the saccade, but these are just averages. And the one problem with averaging when you're looking at diffusion is, as I said before, it suppresses the variance, so you don't actually get to see the variance in the diffusion paths, because the diffusion paths, when you average across them, you just see the deterministic component, the drift. Okay, so we believe that these are representative of pools. If you look at the correlation, the pairwise correlation between neurons, you see that they cluster in positive zone around 0.2 correlation, which is pretty typical from areas of the cortex, like say MT, when you when you get a bunch of neurons that share the same direction preference, or the visual cortex, the primary visual cortex, if you're looking at neurons that share the same orientation preference and so forth. And interestingly, this correlation shows up in the baseline before the random dots turn on too. So we don't think it's an artifact of stimulus, and it might be just a sign that they're connected. Now we can also look at I just showed you those traces aligned to onset of the random dot motion, but here are the individual trials now, but now aligned to the time of the saccade. And again, we think that the saccade might end, or there might be some kind of termination event, something like 80 milliseconds before the saccade begins, the eyes begin to move. And so that termination is what is going to be, we'll return to that when I bring back the colliculus in a moment. But you get the sense that there may be some indication that these individual trials might be crossing some threshold, okay? And indeed, if you just ask what was the firing rate of the neurons in this window drawn in gray, you see that it's the same regardless of the monkey's reaction time, or roughly the same, okay? And that is a pretty good indicator that there's something like a bound going on. And not only that, that the single trial representation of the decision variable, we would argue, is also a reasonable candidate for something that's being sensed in order to terminate the decision. Okay, not so for the other choice, for the reason I just said. Okay, although there's some interesting correlations there, but I won't get into that with you. Interestingly, if we're really looking at diffusion processes, and if you were to say look at the neural response at the beginning of the period of evidence accumulation, it begins just before the 200 millisecond mark. So if you look at each of these times and you try to correlate trial by trial variation in the firing rate with the monkey's reaction time, and we'll stick to reaction times that are greater than 700 milliseconds, okay, that you see this nice inverse correlation. In other words, when the firing rate happened to be a little high, the monkey's reaction time happened to be a little bit shorter, okay? And these are just simple, Fisher Z type tests really t-tests for significance, but don't worry about that so much. Here's an interesting point. So you could write this as a rho is the correlation between the strength of the motion of the firing rate, S of t. This is a mean, right, a single trial firing rate, and the RT. Okay. Now, in this, an interesting prediction is that if what you're looking at is the decision variable, then if you look later in the same trial, and we plot partial correlation, so the same kind of the same times, but take the firing rate and correlate it with reaction time, but conditional on knowing the firing rate at 600 milliseconds or any time thereafter. Then, and for those same RTs, of course, then you see that correlation goes away. In other words, the early correlation is mediated by the later activity. So it's consistent with the Markov process and a common decision variable that begins to show its signature of its relation to the end of the process right from the beginning. Okay, now I want to come back to the main theme and get back to termination. So I showed you this before, the 7 LIP neurons recorded in this session that were recorded simultaneously with neurons in the superior colliculus. And here's what the superior colliculus averages look like. So these are, again, these are like traditional kinds of averages across neurons and trials that we would do. And, it looks like, kind of a squished version of, sorry. It looks like a squished version of what you see in LIP. I just blew it up so you could see. So there's a reason that many of us have thought that the superior colliculus it gets direct input from LIP. That it also represents in these prelude neurons, these neurons that also show persistent activity before an eye movement, that they also represent the decision variable. I'm going to show you that that's incorrect. So I already showed you this type of graph before. And now we look at the superior colliculus and we don't see anything like it. So these are now the single trial averages from the superior colliculus, okay, recorded at the same time as these, okay? And what you see are things that basically don't change much at all for the most part. Most of these trials are just going straight along here. But there's these little bumps that you see, okay? Now, if we look backwards from the time of saccade initiation, we get a sense of what this is. So, okay, so here are these big giant bursts that I'll call saccadic bursts. But you see that earlier in time, in individual trials, I've highlighted one of the bigger ones, you see these bumps, okay? Maybe this is a little bump, so there's not just one necessarily in a trial, but they recur in about on order 15% of the trials in our hands, and I should say in Gabe's hands, and they look a lot like saccadic bursts. So I'm going to refer to them as either premature bursts, or I'll just call them bumps, so we can really tell the difference. Bumps and saccadic bursts, okay? But really, they're probably something like the same kind of process. All right. Now, okay, so if you look in LIP, you don't see something like that. It's hard to do these traces one at a time, but anyway, it's very different. So Gabe devised a method for detecting when these saccades begin, and his method allows us to find the beginnings, when these bursts begin, and whether they're premature or full-on saccadic bursts. And the difference seems to be that they're in their size, okay? So in dark here is a histogram of the amplitude of the saccadic bursts, and in gray, the premature bursts, what I'm calling bumps, okay? There's some overlap, but okay, we're interested in what makes the difference of them. So what we do is we take that starting point and align the activity in the superior colliculus to the beginning of those bursts. So either the premature ones, the bumps, or the saccadic bursts, the ones that lead right to a saccade, or a control where there's no burst, okay? And you see they start out the same way, and the big difference is the size. Fine. Now, remember, we're recording from LIP simultaneously, and in this case from 7 neurons. So these are the 7 neuron averages in LIP aligned to the beginning of the burst in the superior colliculus. And what you can see is that there's a little uptick just before that, okay? So you see that ticker, I've written blue here, and then it looks pretty much the same, this little transient increase in the firing rate in LIP. But and that occurs for both types of the bumps and the bursts, let's say. And the big difference between the two in LIP, the difference between making a burst or not looks like it's a level. So the level of activity, okay? These are normalized and stuff, so we can't read out firing rate. But the idea is that something is acting as a kind of, it's not a pure threshold detector, or we might say the mechanism of detecting the threshold involves some kind of interaction between the derivative of what's going on in LIP, the population of LIP, and its absolute level, okay? And we'd like to understand that better. One clue we have is that if we look at the size of the change, that little transient increase, that the number of bumps. So we're looking at premature bursts. Okay, so these are not near the time of the saccade, but you get more of these bumps when LIP has a larger blip. Okay, a larger transient increase in the response. Okay, and there's the graph that quantifies that. Okay, it's the probability of getting a premature burst, in the next moment, and some short time window that we allow, and as a function of the size of this derivative, basically. I shouldn't have said the size of the derivative. It's really the amplitude of the change. Okay, so that I can summarize. I think I'm out of time, so and I will try to summarize. Well, I'll take my time doing it. Okay, so the first point I've tried to tell you is that LIP represents the accumulation of noisy evidence, and we're seeing that vividly. It doesn't represent, the single neurons don't represent ramps directly. Those are averages of diffusion paths. They don't represent steps. They're averages of diffusion paths. And superior colliculus neurons, they apply a threshold, but they don't represent the decision variable V of t. Okay, so their cross-trial averages actually belie this underlying strange set of bumps or bursts that are premature, that don't lead to a saccade. Okay, so that's kind of interesting. That in both cases you can't infer the underlying process from the averages, but we had inferred a long time ago that the averages of these LIP so-called ramps are actually underlying their averages of diffusion paths. Now, these superior colliculus transients, whether they're premature bumps or saccadic bursts, are always preceded by an increase in the LIP firing rate, and the bursts are also preceded by higher firing rate in LIP. So we suspect. Oh, sorry, I didn't want to say this quite yet. Damn, I hate when I do that. We don't. Well, maybe I've said enough about that. Right. So we think that we might have our hands on something that gives us an idea about what the mechanism might look like in superior colliculus to produce a successful burst. But what we really think is true is that there are other processes that are involved, specifically from the work of Long Ding, Okihide Hikasaka, Bob Wirtz, that the frontal eye field, and especially the action of the frontal eye field, and possibly the LIP, via the caudate and ultimately through the substantia nigra pars reticulata, in other words, the kind of the globus pallidus that projects to a superior colliculus, is also playing a role in the conversion of what would have been a bump to a successful burst. Oh, I did forget to show you one other thing I want to say, which is that two things. I'll just say it here in this slide rather than going back, which is that the superior colliculus, therefore, is playing a critical role. It's the terminator of the decision. Okay. Now there may be another terminator too, for all we know, in the frontal eye field, for example. But you might have noticed that once the superior colliculus makes its burst, then what happens next is the LIP, those LIP aligned to the burst, those responses come down. So something tells LIP, you're done with the decision, stop accumulating evidence, and we think that might be a signal from the superior colliculus. Okay, so ultimately, we think that understanding the terminations in decisions and the accumulation process and their relationship are relevant to problems that we encounter even in neurology, impulsivity, decision policy, speed versus accuracy, which is how we express ourselves as different kinds of decision makers. This is one of the policies that we apply. How we learn, I believe that it's very likely that in perceptual learning, that what changes in the brain are not synaptic weights so much as changes in policy, like accumulating more evidence, and so changing the speed accuracy trade-off. And in some of these, when we start talking about the policy that one uses to make decision, I think we begin to touch on the connection that neuroscience has to ethics, which is, after all, at least from the perspective of contractualism, a non-consequentialist view of ethics that really begins with Aristotle and really through Kant, is the idea that ethics is really something that humans do together to work out how we will behave and what we think is right and wrong. Okay, so with that, I'll stop and thank you, Kheli Mamnoon, and take your questions, and just mention the people that did this work. I already mentioned Gabe and Eric. I didn't mention that Natalie Steinman contributed a great deal to the analysis of the LIP multi-neuron recording data. And I showed you this recently discovered painting of Magritte, an autobiographical painting, a self-portrait of him deciding, should I paint Ceci n'est pas un pipe or shall I paint La Clairvoyance? And here's how he rendered his decision here, and what he decided ultimately to paint is shown here. So that's La Clairvoyance. Okay, thank you.
Hi, Mike. Thank you for the great talk, and thanks for accepting the invitation. So I had a question at the end. So I was going to ask then, we see the kind of a ramp-like activity in SC over trials, but then at the end you explained it very nicely that you think that these are appearing because of the increased probability of those bumps. So within each trial you don't have a ramp up, you just have increased bumps. And I think there are some other people that think increasing bumps can be a evidence accumulation rather than decision variables that rise through drift diffusion. But anyways, so what you showed in one of your slides was that you showed a relationship between the probability of these bursts, these bumps, the premature bursts, and the derivative of the firing in LIP. So which still, I think in my mind, brings up the question that we all thought that when LIP firing reaches a particular threshold in firing, that causes a saccade to be generated. Now, the plot that you were showing was more like it's the local change of the fire. It's the derivative of the firing that is causing a saccade. How can you reconcile these two different views? Isn't it a bound that you have to reach absolute value of firing?
Yeah. Yes. Okay. So I think you're there. Maybe I was not clear enough. So I shouldn't have called it a derivative, by the way. I just should have said the size of that brief increase, okay? Because actually the slope looks very similar, okay? So, but if it's a bigger excursion in some short time, then you're more likely to get not termination, but a bump. So it's like the superior colligus. The way the threshold mechanism works is it needs two things. It needs a positive going step. Okay, that's obvious. The last step should be a positive going step, right? And it needs to sit on a higher firing rate. And those two things together could be used to produce a threshold mechanism that looks like it's operating on a threshold level in LIP. Okay, so that might be all it takes. We don't think that's really true. We think there's something else going on in LIP in superior colliculus. We think that release of inhibition from the substantia nigra pars reticulata might also play a role, and it might play that role in part because the frontal eye field or some other structure is sending a signal to the substantia nigra pars reticulata via the caudate. You follow my reasoning here? So I haven't abandoned the idea of the threshold. They're completely compatible. Okay. Now the other thing you mentioned is you mentioned two other things I want to address. One is that the bumps. We know that the bumps account for a lot of what we see in the averages, but we're not sure that they account for all of it. Okay, so there might be a rumble that is ever so slight that in the activity in the superior colliculus that's also preparing it to be able to accept this little change from LIP into a burst instead of a bump. Okay, and the bursts are the ones I'll use the burst just for the saccadic bursts, the bursts that lead to an eye movement and terminate the decision. Okay, so the other so we don't know though that accounts for all of the average that I showed you that looked like decision variable. Okay, we think it accounts for a good fraction of it, but we haven't nailed this yet. And then the other thing you mentioned is that yes, some people think that you can explain the ramp activity in LIP through the averaging of they didn't say bumps, but steps, okay? So a step to a high level or a step to a low level. And we think that we never thought that was correct, and now we get to see directly from very small populations of weakly correlated neurons that we see diffusion. I think it's very hard to, we're working hard on trying to see whether there's any way to save a stepping model, but I think there isn't.
Okay. Yeah, and I mean, I think that of course the design is great, and since you can record simultaneously from neurons that share RFs, I think this is the way to go. But as you also mentioned, it's still not clear how the firing of the LIP is translated into the decision of the superior colliculus to initiate the saccade. We have some clues, but it's still in my mind not clear who sets the threshold. How does the threshold set? How is this threshold set in the superior colliculus? So
I recommend that you read papers by Tiago Branco. Uh-huh. Okay, so he has beautiful papers in the mouse doing these escape behaviors. Okay, A couple of them that are interesting, but the one that I think gets into the mechanism is his first paper, which has to do with what he thinks is integration of a looming signal that leads the mouse to either freeze or run. And you'll see how he does it. When we build these mechanisms, we will write them down as threshold detectors, but they're actually just something that's a very highly nonlinear dynamical system that is under the control of something else, say, again, the substantia nigra pars reticulata, that allows it to go into this sort of explosive burst. And that's all you really need, so long as the explosive burst can then say to LIP, stop integrating.
Okay. Well, thank you.
I've got a question. If we assume that the superior colliculus is responsible for terminating the decisions, I was wondering how much or what proportion of neuron you recorded have this bump?
Right. We don't. Good question. So we're focusing on the prelude neurons, and and so some neurons are pure burst neurons. Okay, they don't show any prelude activity, meaning they don't have like persistent activity when you plan an eye movement. And so whatever that fraction is in the intermediate and deep layer, and I'm guessing it's a pretty large fraction. But what's missing from here are, for example, the direction selective neurons. We have had some of those that are in the intermediate and deep layers. Intermediate, not the deep. So we don't know what they're doing. They were reported by Horwitz and Newsom many years ago. They interest us mainly because of the pathway from the colliculus to LIP through the pulvinar. And so it's a large fraction. You might be interested in knowing this, though, is that when those prelude neurons are active in a delayed saccade, like I showed you, like either by memory or even just overlap, where you just wait while the stimulus is on, then we don't see any of these bumps.
Hi, thank you for your talk. So my question may not be directly related to what you presented, but more on the cognitive side. Do these bumps, not the bursts that you see in the colliculus, but the bumps that are happening before the burst, could they be related to the conditions when the monkey is not certain about what is happening or what choice he has to make? Or can you comment on that? Or is there a way to test this?
Well, so we have done lots of experiments on certainty, confidence, things like that. He's not certain enough to answer with an eye movement, so we know he's not that certain, at least up to the degree that he feels he needs to be certain, okay? And we're applying cognitive words onto something that is just a process, right? But that's fine. But we know how to interpret those decision variables, which are diffusion paths. We know how to interpret them in terms of the certainty that they should and do confer, meaning there's a mapping between the height of the decision variable at any moment in time and the amount of time that's elapsed and the probability that a decision based on the sign of that accumulation will be the correct one. Okay, so that's what Rusbeck and I defined as our meaning of confidence, which is there are lots of uses of the word confidence, but I think that's a useful one. And so no, I don't think that you could say that any positive step that begins on a positive baseline is an increase in your confidence. But I'm tending to think about this just more mechanistically. We don't know what do we do to control the speed accuracy trade-off. Now, and for years we've been noticing that there's a signature of something that's looking at a threshold in LIP. Okay, or again, something that's highly correlated with that in another area. But let's just say looking at that threshold in LIP. So what sets that? Well, okay, one thing I should say is that threshold is constant. Okay, it's constant in time, as I showed you in that graph from National, but it's also constant across conditions like differences in speed accuracy trade-off. So that means from that we infer and actually showed that the way you control your policy, your speed accuracy trade-off, is you control it not by changing the bound, but by changing the starting point of the accumulation.
Right.
Okay. And similarly, there's a reason that the bound ought to collapse in a time-dependent way. And the flat bound I showed you, that consistent firing rate level at the end of the decision, it is not an argument against that. It's just that the way it's implemented in the brain is an additive signal to the accumulated evidence. It's another determinate signal. Okay.
Right.
So but we'd like to understand it at a mechanistic level because, you could imagine, I mentioned a term that has clinical significance as well as cognitive, to use your word, and that is, things like impulsiveness.
Right. Yeah. Yeah. Thank you.